Welcome to this webcast, which is part of our series on antimicrobial peptides. With me today is Dr. Keith Miller. He's a senior lecturer of medical microbiology at Sheffield Hallam University. Keith, it's great to have you on. Thank you so much for being here.
No problem at all. Happy to be here.
Keith, antibiotic resistance is one of the main challenges we're facing, and the pressure to find new options is increasing. Can you first share with us maybe where your current research aims to find new solutions for this challenge?
Sure. So my background before I started working on antimicrobial peptides was in traditional small molecule antibiotics. But as we all know, and I know nobody wants to talk about the next pandemic whilst we're still in the midst of the current pandemic. But as we know, the problem with antibiotic resistance is increasing all the time. By most estimates, there's around about 750,000 people a year that are dying of antibiotic resistant bacterial infections. And so myself and many other people believe that working on small molecule antibiotics, whilst important, is just likely to result in similar problems in the future. We know that antibiotics are incredibly widely used, but we know that the problem of overusage is resulting in the problems of antibiotic resistance. So myself and other people started to think about, well, what could we do as an alternative? And antimicrobial peptides are found in all forms of life on the planet, from bacteria to plants to mammals. We create antimicrobial peptides ourselves when we're exposed to pathogens. And so we figured if nature thinks that this is a good idea, then it probably isn't too bad an idea. So we started developing our own antimicrobial peptides that are derived from those peptides that we find in nature, in part to combat the antibiotic resistance issue. Antimicrobial peptides target multiple sites on the bacteria rather than just a single lock and key type mechanism, which means although resistance is possible, and we do see resistance, unfortunately, to antimicrobial peptides, it emerges much more slowly. It comes at a much greater cost to the bacteria themselves, so they grow more slowly. They're unable to exist and thrive in the same way once they develop these resistances to antimicrobial peptides. So evolutionarily speaking, it's disadvantageous for them. So antimicrobial peptides are a good candidate in terms of avoiding some of the more simplistic elements of antibiotic resistance. So that's our thinking behind developing these as alternatives to traditional antibiotics.
Great, thanks so much for that. And do you think there's a particular reason why we're finding lots of antimicrobial peptides in snake venom?
Well, so the reason we look in snake venom, it seems a counterintuitive place to look for medicines. I completely understand that. Snake and scorpion venoms are obviously ancient products. You know, snakes and scorpions have been on this planet a lot longer than we have, but they have a much less sophisticated immune system. So snakes and scorpions only have what we call an innate immune system. So this is when a pathogen comes in and you get an instant non-specific response to dealing with that pathogen. We have that response as well. It's what makes us feel dreadful when we're exposed to the coronavirus or to the influenza virus, or we get tonsillitis and we get those headaches and aching joints and muscles. That's because we're seeing this non-specific inflammatory response. The difference between us and snakes and scorpions is that during that period of time, we then have what's called an adaptive immune response. So where we see antibodies produced, where we see T-cell responses. Snakes and scorpions don't have that. They only have the non-specific response. They also have quite simplistic circulatory systems. So whereas we have blood and lymphatic circulatory systems, snakes and scorpions have a combined thing called hemolymph. So this is where the immune molecules and the normal kind of oxygen carrying molecules are mixed together and circulate around their bodies. The supply to parts of their bodies aren't fantastically great. Now, snakes have a venom gland close to their fangs, and scorpions have a venom gland in the telson, the stinger on the end of their tail. that doesn't have a very large supply from the hemolymph. So if they were relying on things being absorbed across from their hemolymph into those glands, it would take a long time for that tissue to become protected from pathogens. Obviously, those two things are completely essential for these animals. They rely upon their venom for hunting, so they rely upon it for food. If their venom gland becomes infected and unusable, they'll starve to death. Probably more quickly than that, however, they rely on those things for defense as well from predators. And if those predators can no longer be protected against, then they'll die because they'll be eaten effectively. So the venom gland is really, really important. It's also, unfortunately, very, very exposed to environmental contamination. If you imagine a snake in the desert eating a gopher, the fangs go down into that gopher, they could puncture that gopher's bowel. There's all sorts of bacteria, potentially pathogenic bacteria in that environment. Fangs are just a hollow tube. The bacteria can travel up that tube, access the venom gland, cause an infection. Clearly, the neurotoxins in venom that affect human beings are completely ineffective against bacteria that have no central nervous system. The way venoms attack us is by attacking our nerves or perhaps clotting our blood. Bacteria have no nerves, they have no blood, so those neurotoxins have no effect. So our idea, not just our idea, I mean, we looked at this throughout the literature, is that there must be something else in the venom that protects the producing organism from invading pathogens. In snakes and scorpions are really ancient, so there's about 150 different types of molecules in scorpion venom. In snakes, it's more like 100. 2 or 3 of those are the venom toxins, and everything else is there for some other reason. And so our idea was, well, at least 1 or 2 of those are probably there to protect the venom-producing organism from pathogens, and that's what we were looking for.
To get to broader scale adoption of antimicrobial peptides as a potential alternative to more traditional antibiotics, what challenges do we still need to overcome?
Yeah, quite a few, unfortunately. So obviously these molecules, we can't farm millions or possibly billions of snakes and scorpions for access to antimicrobial peptides. That makes absolutely no sense. So the first thing that we have to think about is how can we make these antimicrobial peptides in a different way? A lot of antibiotics and a lot of drugs more generally are made by taking the gene from whatever organism produces it, putting that into bacteria and growing them in giant vats and then extracting the peptide from there. There's a problem with that, obviously, with antimicrobial peptides, in that clearly the thing that we're looking to produce would kill the bacteria that are producing them. So most of the time we're looking to make these things synthetically. So we take a combinational chemistry approach where we try and make these peptides in the laboratory rather than extracting them from the producing organism. That's relatively straightforward because we know the sequence of these antimicrobial peptides, but it's quite expensive. So obviously there's an economy of scale. So the more you make, the cheaper it becomes per unit. But we still need to think about ways of making it cheaper. The other problem is these antimicrobial peptides are not very what we call drug-like. So although they have a good effect on the bacteria within a test tube or an agar plate, in terms of when we would put that into a human, we have to get it dissolved into the bloodstream. We have to have it be transferred around the body and diffuse into tissue. We have to have it not be immunogenic, so people don't have allergic reactions to it. It also needs to exert its effect quickly. And because these are big, bulky molecules, that's not always very straightforward. So, as well as looking at ways of making these things synthetically, we're also looking at ways to make them more drug-like, to shrink them, to make them smaller, to give them more drug-like properties, to make them less likely to bind to serum proteins in human beings' blood, where it can then no longer diffuse into tissue. So there are a number of challenges. We need to make it more like a drug. We need to make it less reactive and less toxic to humans, whilst at the same time retaining its antibacterial activity.
And if we look beyond antibacterial activity, what other areas do antimicrobial peptides have potential for, or where could they be useful as well?
So we already have antimicrobial peptides from other sources that have anti-cancer potential. So the way in which antimicrobial peptides work in terms of targeting bacterial cells is, in the first instance, they have a charge on them, which is attracted to the membrane of bacterial cells. What some people don't know is that cancer cell membranes behave very differently to normal human cell membranes. So they carry themselves a different charge to the cells that are found normally every day in human beings. As a consequence, charge-attracted antimicrobial peptides can actually be directed specifically towards cancerous cells rather than normal native cells. There are lots of antimicrobial peptides that work against viruses as well as against bacteria. So we're able to identify antimicrobials that have a spectrum of activity that goes beyond bacteria and so can target viruses. And already antimicrobial peptides have been used very successfully in that field. Looking beyond antimicrobial peptides, there are loads of things in venoms that have been used as antihypertensive medications, anti-inflammatories, pain medication. So venoms really are a treasure trove of potential medicines.
And so where do you see now the near-term future for antimicrobial peptides? What will happen next? And can you maybe also share a little bit what you are working on?
Yeah, I think the depth of research now in antimicrobial peptides is greater than it's ever been. And we're seeing more and more groups working on them. We're seeing more and more publications appearing in the literature. So I think, you know, there's a critical mass building in terms of developing antimicrobial peptides for clinical usage. And I think we'll see more and more of them appear in the clinical sphere over the next kind of 5 to 10 years. In my own research, we've largely stopped prospecting for antimicrobial peptides. We've stopped looking at more and more venoms from more and more diverse animals. I mean, we've looked at all sorts of things. We've looked in snake venom, scorpion venom, siderophore venom, bloodworm venom, biting insect venoms. I mean, we've looked at a lot of places, and we've got a lot of good candidates out of that. So now we're focusing our efforts on making them more like drugs. So we're looking at truncations, so that's making them smaller. We're looking at amino acid substitutions. So this is where we're trying to change the properties of the antimicrobial peptide. And we're looking at ways to scale up synthetic manufacture as well. So we're now largely focused more on the kind of end stage drug discovery and are optimistic that that will result in some drugs in the future. As well as that, we're interested in the secondary effects of antimicrobial peptides. So most of our research has been focused on the binding of these peptides to the membrane of bacterial cells. But one of our recent publications identified that there may be intracellular targets for these antimicrobial peptides. And so we're investigating the kind of the dual mode of action of these antimicrobial peptides. This will be really helpful in drug modification because at the moment, all of our modification efforts have been around being more efficient membrane disruptors and haven't paid any attention really to a potential secondary mechanism of action. But if we're able to figure out what that second mechanism of action is, we can start to modify the drugs to enhance that activity as well, to make them even better as clinical drugs.
Keith, thanks so much for all this information, and best of luck with your further work. Thank you.
Thanks very much.